Okay, hello everybody. We're going to talk a little bit about why nanopore sequencing for plasmids is better. So, DNA is the language of biological systems, and if you want to interface with these systems, then you need to modify DNA somehow, introduce it to them. And the most common way that this is done is using a plasmid. Plasmidsaurus was the first company to bring a nanopore-based plasmid sequencing service to market, and has been extremely successful because it is unequivocally the better way to do it. Please allow me to explain. Almost 50 years ago, in 1977, Fred Sanger published his method for sequencing by synthesis, building on the principles of PCR, which is without a doubt one of the most impactful technological innovations of the 20th century. And a fun fact to get a sense of how long ago that was, 1977 was also the year the first Star Wars movie was released. So the way this method worked is that you would use the restriction sites in your plasmid to cut out the insert that you had inserted, amplify it, and sequence it. And what this allowed you to do was confirm the sequence identity of that insert. So you could make sure that you were actually inserting the gene that you thought you were inserting. So this is obviously an extremely important QC step that can save you a lot of time downstream, because you can identify, like, really early on in the process, any potential issues. But half a century of data have shown us that this method has got some problems. So Sanger sequencing relies on DNA polymerase, which is an enzyme with a couple of quirks. For example, it really struggles with hairpins, high GC, and repetitive regions. So you can see here on the left that the sequencing reads simply stop. So this is real data visualized in Snapgene. And this is an example on the left of an ITR highlighted in green. And then on the right is an example of a repeat. So you can see the sequence at the bottom. And these are the in red are the reads that have been mapped to it. So where it's empty instead of red means that it couldn't map successfully. So you can see from that these reads basically cannot align during this section, sort of in this section. And that's because the polymerase basically can't resolve the hairpin, and it can't make sense of the repeats. And this is not polymerase's fault. This is just not what polymerase is good at. And moreover, so because Sanger sequencing as a technique is based on amplification, this can paper over problems. So as an example, if 10% of the plasmids in your sample, like in the actual tube, are properly formed, with Sanger sequencing, your primers would bind to it, it would amplify it, your results would look great. So you would say, I have good sample, this is what I expect it to be. And then you would do your downstream experiment and it might fail. And we see that a lot of the time this wasn't what was happening. And it's because the QC test of Sanger isn't actually a very good QC test, right? It's not what you see in the results, doesn't reflect the truth, which is that only 10% of the plasmids were fully formed or properly formed as you expected. And if we look here, so again, these are real reads, these are nanopore reads. You can see they read all the way through and they align the whole way along. So another advantage is that there's no read length limits. You probably noticed that they were significantly longer in the previous slide. Sanger reads have a limit of about 1,000 bases. So if you can see along the bottom, this plasmid, these numbers represent the length. So it's kind of maybe 7 or almost 8,000 bases long. And Sanger reads have got a limit of 1,000 bases. So it means that if you want to sequence the whole plasmid, you'd actually have to sequence it multiple times. And it's possible to do, as this image shows. So this is, again, real data. But it's very annoying because you have to design primers that ever so slightly overlap. And you still have to make sure, all of the normal primer things. You have to think about melting temperatures, primer dimers, primer hairpins, GC content of the primers, which obviously just takes a lot of time. So it's possible, but laborious. And then not to mention that doing multiple Sanger runs actually becomes quite expensive. So if you were to do it this way, it would cost significantly more than doing it with us. And so with Plasmidsaurus whole plasmid sequencing, you would never need to design primers again. And to be clear, that's not because we design the primers and we just make other people not have to design the primers. You just don't need primers. Primers are not needed. And you can see here again that this is a nanopore read. It just reads right the way through the entire plasmid with no issues. And Sanger sequencing does not work if you have multiple plasmids. If you've got a mixture in there, or even, and this is again another annoying thing about primers, if you have multiple possible binding sites, what will happen is the primers will bind up both or multiple of the sites, and then the sequence is no longer the same. So it looks exactly like this. This is a real Sanger trace file. You get this overlapping peaks and mixtures. And if you look at the top where it should tell you what the base is, you'll see it's an N, and the N means the software can't tell you the base. And it's because Sanger isn't a single molecule technique. Everything is lumped in together. So Plasmidsaurus whole plasmid sequencing, the way we do it, has got no issues with any of these things. High GC, repeats, hairpins, inversions. It sequences right through them. And that's because there's no polymerase. So you can see here on the right, we make use of a modified DNA helicase. So that's represented by this little donut just sitting above the nanopore. So this obviously unzips the DNA, and it also crucially controls the speed at which it moves through the nanopore. Because if you did it without the motor protein sitting there, then it would zip through at something like 1 million bases per second, which is too fast for the sequencing algorithms to make sense of the sort of raw data squiggle. The base calling algorithms. And so the helicase as an enzyme was designed specifically, evolved to get through tricky secondary structures. It actually just breezes right through even tough ITRs. And like I said earlier, there's no read length limit. So above the pore, you can see there's DNA that could actually be of any length. And the current record for the longest read is over 4 megabases, which is almost an entire E. coli genome in a single read. And so as Sanger is limited to 1 kb, people usually just sequence a portion of the insert. But the way we do it is you get this full picture. So you get information about the insert, but also the whole backbone. So we use a transposase, which will basically randomly cut somewhere around this circular plasmid DNA, and then the sequencing adapter is added where it cut. And so what you get is a full-length read starting at a different point. And then every day, literally every single day, we hear from customers that they finally understand why their experiments were failing. So they did it the right way, they did the gold standard, and then their experiments failed, and oftentimes they just gave up forever. Other stuff took priority, and they had to move on because it wasn't working. And it's because they were missing all of this critical information. So you can see in this example, everything had concatemerized, which is something plasmids are wont to do. And in this example, there's a big insertion between the kanamycin resistance gene and the origin of replication. And because it works at the single molecule level, which means that every individual piece of DNA is read separately, it's really easy to tell a mixed sample. So dimers, trimers, these concatemers are easy to tell because you see multiple peaks at exactly 2x and 3x the length. And this is what it looks like. So this is actually better than running a gel, because what happens with a gel is if it's concatemerized, the restriction sites also get duplicated. So you might cut it out of the plasmid, run it on a gel, be like, great, it's the exact length I think. But what might have happened is you might have pulled 3 inserts out from actually a single plasmid, and the gel would only show that one band. And when you sequence them with this single molecule platform, what you see is the truth, right? So you easily can tell if it's concatemerized, if it's 2x, 3x, you can easily tell, as you see here, 2 distinct plasmids, and you can tell if it looks great. And also because of the single molecule nature, if only 10% of it looks right, only 10% of the reads will look right. So it gives you a much better sense of what's really in there. So to summarize, you're getting the entire plasmid, not just the insert, and the single molecule nature allows you to really see what is in your sample. The sequencing is also extremely fast, and best of all, there are no primers. There's no primer design, there's no primer walking. You just send it to us, we snip it open with a transposase, and sequence a whole thing. So hopefully you agree with me now that there is really no question that this is the much better way to sequence your plasmids, and you should not be sequencing them with Sanger anymore. Thank you.